Okay, so this week we're talking about topic 1.6, which is all about cell division. All right, so the first question is probably why do cells need to divide at all? So if you think about it, you came from a single cell, and that one cell divided into the trillions of cells that made up you today, right? So one reason is development. The other thing is repair, right? So every time that you get a scratch or you break a bone, or if you're this unfortunate lizard right here, he lost an entire arm, we need cells to divide in order to repair our tissues when they get damaged. And then the other reason is just like straight growth, right? So you're a lot larger now than you were when you were a little kid. And in order to do that, we need to have cell division. So cells divide in a really specific sequence of events that is known as the cell cycle. So we're going to be learning all about this today. So cell division is going to produce two genetically identical cells. So this is different than meiosis when you were producing those four haploid cells that had that mixture of different chromosomes. And the cell cycle is divided into two main parts. So interphase, which is the longer of the two and consists of these three subsections, G1, S, and G2. And then the second part of the cell cycle is going to be mitosis, which is going to seem really similar to meiosis. So if we ask the question, what's all the stuff that makes up a cell? Like, what are all the different parts? And so you know from 1.2 that the cells have the phospholipid bilayer, they have cytoplasm in the interior of the cell, the cells have a nucleus, they're going to have different organelles, and if I want to divide, I have to go ahead and make enough stuff. I have to double it if I'm going to divide it into two cells in the end. So to do that in the different parts of the cell cycle, the first part of interphase is going to be G1. So the G1 stands for gap 1. And what's happening in G1 is you're getting a lot of protein synthesis because remember, you're trying to double the cell size. The other thing that's happening is all your organelles, your mitochondria, your ER, the Golgi, all of those numbers are going to be increasing in size and number. The cell membrane is also going to start to expand. It's going to increase in size. So that's G1. So the next phase of interphase is S phase, and that stands for synthesis because DNA is being replicated during that S phase. So these are still considered diploid cells even after they replicate because chromosomes are counted by the number of centromeres. So when this DNA replication takes place, it's going to result in sister chromatids. So when these are genetically identical, when they replicate, they're called sister chromatids, and they are identical, and they're going to be attached to each other by something called a centromere. And so again, when they're counting chromosomes, they're only counting by centromeres, so we're still considered to be in a diploid state during S phase. And the most important thing about S phase is that DNA is replicated, creating those genetically identical sister chromatids. So the next part of the cell cycle is going to be G2, which is gap 2. And so just like in G1, it's kind of a repeat. You get even more protein synthesis, even more organelles that are going to be increasing in number. And again, that cell membrane is going to increase in size, and that cell is now 2 times the original size because it is about to start dividing at the end of G2. So both G1 and G2 are have these things happening. So the protein synthesis, the organelles increasing, as well as the cell membrane. So these three parts of the cell cycle together make up what is called interphase. So interphase includes G1, the S phase where that DNA synthesis is happening, and G2. It's considered the longest part of the cell cycle, and it is marked by growth both in the cell size and in DNA replication. So it is basically doubling everything in preparation for that cell to divide into two. So the last part of the cell cycle is M phase, and so that is for mitosis. And mitosis is all about producing two genetically identical nuclei. So if I ask you what mitosis is, this is the answer that you're going to go for, two genetically identical nuclei. And mitosis, just like meiosis, is going to be following the separation of some chromosomes, but in this case, instead of following the separation of the homologous chromosomes, we are now looking at the separation of sister chromatids in mitosis. So it's going to have these 4 stages that should sound very familiar to you: prophase, metaphase, anaphase, and telophase. So these are the same ones that we saw in meiosis. It happens just once in mitosis. So let's take a look at these different stages of mitosis. Remember that mnemonic PMET, right? So this is going to give you the order of the different stages of mitosis. So if we start with prophase, and I need to get rid of that because it's not prophase 1, it's just plain old prophase. In prophase 1, we have the chromosomes that are condensing by supercoiling, and this is where they first become visible. So the supercoiling is where the DNA is wrapping around those histone proteins, right? So that is occurring in prophase 1. The other thing that's happening in prophase 1 is that you have that nuclear envelope start to break down because it's going to need to separate these guys. And then you also see the formation of spindle fibers that start to form and attach to some of those centromeres. And all of this is occurring in prophase 1. So for mitosis, you will be responsible for knowing more deeply what is occurring in each phase as compared to what you were asked to do in meiosis. So know that this is all occurring in prophase 1. What is not happening here is any kind of crossovers between homologous chromosomes. That is only in meiosis. So for your similarities and differences section, the crossovers only happen in prophase 1 of meiosis. Okay, that does not occur during mitosis. So the next part of PMAT is the M, which is metaphase. So that's when the chromosomes, the sister chromatids, are going to align up along the middle of the cell. So that's metaphase. The other thing that occurs is that the spindle fibers are going to attach to the centromeres on each sister chromatid because they're about to separate them, right? Because in anaphase, those spindle fibers are going to start to contract, which is going to separate those sister chromatids. So anaphase, it starts with A, they're moving away from each other. So now I am no longer diploid. I've moved from a 2N to a 4N state because each of these guys still has a centromere. So now I have 4 times as many chromosomes as before. So the last stage is telophase, and it's going to be in a 4N state until that new cell wall or cell membrane forms. So in telophase, the chromosomes are going to move to opposite poles, and then that nuclear membrane is going to start to reform, and you'll have cytokinesis actually begin, and they will remain in a 4N state until that those new membranes are formed. So just to review, so this is prophase, so you can see the chromosomes start to condense, and then they're going to line up in the middle during metaphase, and what's next is anaphase. They're going to move away from each other, and in anaphase you can kind of see the tails hang out as they're drawing away, and in telophase they sort of start to coalesce into more recognizable nuclei, so that's telophase. So for this next bit, if you did not attend the live session, be sure to answer these questions in your self-paced Nearpod. So what stage of mitosis is this first cell right here of the PMAT? What is that? And then what is this stage? What stage is this cell in? Okay, and then go ahead and tell me what this stage is here. And then, yeah, we'll stop there. Or, okay, tell me what this stage is right here. So this cell right here, what stage do you think that's in? Okay, so that was mitosis. So I had a new word in there that maybe you haven't seen before, cytokinesis. So cytokinesis is the final step in cell division. So this is where you have the formation of a new cell membrane and or wall. If you're a plant, right, you have both. And this is the part where the cell physically divides into 2, but the process is different depending on whether you're looking at a plant or an animal cell. So we'll look at the animal cell cytokinesis first. So let's say I have my animal cell and I'm at telophase where I have my, chromosomes are separated on opposite poles. So what happens in animals is that you have this formation of a contractile ring that's made of microtubules that's being pulled shut by 2 other proteins called actin and myosin. So these microtubules are going to form a ring around the center of the cell. This has a special name. It's called the cleavage furrow. And so what happens is it's like a tie, like a necktie, right? That's made out of microtubules. And that necktie is wrapped around the cell, right? Say it's like a balloon. And then the actin and myosin are going to pull in, they're going to tighten that necktie and form that cleavage furrow. And when it contracts, it's going to pinch off the two cells and separate them from one another. So that's what's giving you the two cells in animals. So this is just a look of what that actually looks like in real cells. So this is cytokinesis happening in animals. We have this contractile ring forming in the center here, and it's being pulled by actin and myosin to contract until it physically cuts off the two cells from each other. They pinch off, and now we have two independent cells. So just another look, that contractile ring's forming, and it will eventually separate these two cells. So again, just one more time. Contractile ring forms in the center. That actin and myosin is going to pull it shut until it completely pinches off into two new cells. So that's cytokinesis in animals, but let's look how it happens in plants. So if I have my plant cell, again, I'm in telophase, I have my nuclei on either side. So in plants, the Golgi is actually involved in this. So remember that the Golgi apparatus packages up things into vesicles, and it's usually sending them outside the cell or to the cell membrane. So it's basically doing its same job. It's just it's making a new membrane, right? So that Golgi apparatus is making vesicles, and remember on the outside the vesicle is phospholipid bilayer just like the regular cell. Only this time, inside the vesicle, it's also carrying proteins like pectin and cellulose, which are going to make up the new cell wall and cell membrane. So the Golgi starts making those vesicles and starts sending them to the center of the cell. And those phospholipids can fuse together and release their contents. And then eventually you form something called a cell plate. So cell plates only occur in plants, never in animals. That's cleavage furrow in animals. And once they fuse, that will become the new cell membrane and cell wall. And so this is how cytokinesis occurs in plants. So this is just a video. This is a real plant cell that's undergoing the final stages of mitosis. And if you look closely, you're going to be able to see the vesicles budding off from either side and forming that new cell wall. Okay, so we're in anaphase. They're going to start moving away from each other. So you see these little vesicles that are coming off, from the main part of the cell, and you see a new cell plate forming, which is going to become the cell wall and cell membrane in the plant, it's a very different process than what happens in animals. So you do have to be able to distinguish between that. Okay, so the next thing on the to-do list is the mitotic index. So this is actually a pretty simple math problem compared to what you guys have seen before. So all mitotic index is the number of cells in any stage of mitosis over the total number of cells. So how these problems are going to go is they're going to show you a picture of some sort of cell sample. And they're going to say what percentage of those cells are in mitosis. So that is the mitotic index. It's the percentage of cells that are in mitosis. Okay, so if I have this picture here, to get that number, I would first count all the cells. So let's see how many cells we have. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34. So I think there's 34. So I would put 34 total numbers of cells here. And then I would go back to this picture and I would count how many cells are in mitosis. Like, what do you think? So I will let you answer that question in your Neuropod. Okay, so for this one, basically do it again. Count the number of cells that you see in here total. And again, you're going to put that number on the bottom. And then you're going to go back and count the cells that you think are in mitosis and put that number on the top and give the percentage of cells that are in mitosis to give you your mitotic index. All right. So, cell division is a pretty heavily controlled process because you can't just have cells dividing all the time willy-nilly. First of all, you don't want to be growing if conditions are bad. So say you're a plant and there's no sun or no water. You don't want to be wasting your resources on cell division when there isn't enough resources around to support that. The conditions have to be just right to promote cell growth. And when we have out of control cell division, it leads to things like cancer, which we'll talk about in a little bit. So how do we regulate the cell cycle? Well, there's a special class of proteins called the cyclins whose whole job is to control when cells actually divide. So you'll be seeing this graph a lot, and you'll have to pick out, who's who, what phase they're in, and everything that they do. So cyclins are proteins that regulate the cell cycle, and there are 4 of them that you need to know: cyclin D, E, A, and B. And I didn't lose my mind. I have them in this order for a specific reason, because this is the order that they are actually expressed during the cell cycle. And you do have to know that. So, make up a mnemonic. I just call it DAB in my head. And so different amounts of each of these cyclins are expressed during different parts of the cell cycle, and that is how the whole thing is controlled. So if I have my cell cycle here, and you're free to either annotate the diagram that I have in your notes, or you can write it in the table. It's up to you. Remember, we start with G1, which is gap 1, where we have all that protein synthesis and the organelles are increasing in number and the cell membrane is increasing in size. So it's actually cyclin D that triggers the cell to enter from G0, which is just it's like on standby. It's not dividing. So cyclin D triggers entry into G1 of the cell cycle when it starts to be expressed. Towards the end of G1, we have the arrival of cyclin E. So once cyclin E starts being expressed, it's preparing the DNA for replication that's going to happen in S phase, that synthesis phase of the cell cycle. Towards the end of S phase, cyclin A is what actually activates that DNA in order to be replicated. And then at the end of G2, just before the entrance into mitosis, we have cyclin B, who's going to actually promote spindle formation. So we need that to separate the sister chromatids. So you do need to know the order of these cyclins, what they actually do, what they trigger. Remember, cyclin D is in G1, triggers entry from G0 to G1. Cyclin E prepares for DNA replication in S phase. Cyclin A is going to activate that DNA replication. And cyclin B is what's promoting the spindle formation in mitosis. So if I were to graph these in G1, remember that cyclin D are the first ones that you're going to see the levels increasing, because this is what first triggers the cell to enter into G1, into the cell cycle at all. And that just prior to the start of S phase, we see cyclin E come on the scene, which is going to prepare that cell to enter S phase. And then before we exit S phase towards the end, cyclin A is what actually triggers the replication of DNA. And then as we enter G2 towards the end, you will see an increase in cyclin B, which is promoting those spindle formations. And then as you exit the cell cycle, the levels of all of them are going to drop off. So again, cyclin D triggers entry into G1. Cyclin E is what's going to trigger the entry into S phase. It's getting the DNA ready for replication. And then cyclin A is going to actually trigger that DNA replication, and it's cyclin B towards the end of G2 is going to ensure that those mitotic spindles are actually forming. So I said at the beginning that uncontrolled cell division results in cancer. And this is a pretty common disease, and most of us know someone, who has been affected by that disease. It's pretty common. It's the second leading cause of death in the United States, and you can expect to have a lifetime risk of that. It's about 1 in 3 or 1 in 4 people who will come down with one of these cancerous diseases. And it affects many different tissue types. So like lung, stomach, colorectal, liver, and breast cancer are the most common ones. And all of these are basically uncontrolled cell divisions of different types of tissues. So let's go ahead and look at this sample here. And you can tell me what sort of tissue do you think this might be, because you actually have seen it before. I just want to point out, so some of these look like little hypertonic cells. Okay, and then what is different? So what sort of tissue do you think this is? So this is a blood smear. So these are red blood cells that you see here. And then these guys are something new that you may not have seen before. So these are white blood cells. They're also known as leukocytes. So on the left side, this is a normal blood smear. And on the right side, this person has a disease called leukemia, which is the uncontrolled division of the leukocytes, the white blood cells. And so just looking at this, you can see that there's some big differences between these two. So first of all, not only are there many more white blood cells, but they're weird, right? They're huge. The nuclei is weird. Like, look how oddly shaped these nuclei are. And so these are just some other images of leukemia blood smears. Again, there's like way too many white blood cells in here, especially for somebody who doesn't have an active infection. And again, the cells look really strange. So this is probably one of the easier ones to diagnose by a pathologist. So that's their job. They look at blood smears and they can say like, yeah, this person definitely has leukemia. So this was one of the leading causes of childhood cancer deaths. It is much more survivable now. I think something like 90% pull through. So cancer cells will divide uncontrollably, and sometimes that can go on for decades or kind of indefinitely. So I want to tell you a little bit about the HeLa cell line. So this is Henrietta Lacks, and she unfortunately got cervical cancer in 1951, from which she died of that at the age of 31. And this is some of her cells known as the HeLa cell line in labs today. So what happened was the doctor that was treating her in 1951, it was a Dr. Gray at Johns Hopkins, took a sample of her cervical tissue and started to grow it in like a petri dish at the lab. And even though she died in 1951, her cells have kept dividing in the 70 years since then and are in labs all over the world today. So this is a real video of her actual cervical cancer cells. So these were derived from her. And even just looking at this now, they're dividing kind of rapidly even on video. And it's estimated that since that tissue was removed from her, it's grown about 50 million tons of HeLa cells that have been grown in all the labs all over the world to date. So this is a pretty extreme example of what cancer is. It's again just the uncontrolled division of cells that can go on indefinitely. So these cells will probably be here long after we're gone. So remember that, so how do we get there, right? How do we get from normal, healthy person to a situation where these cells are dividing uncontrollably, right? So let's go back to our cell cycle. And we know that we have these 4 cyclins, D, which is expressed in G1, E, which is getting ready to go into S phase, and A, which is triggering that DNA replication, and then B, which is doing those spindle fibers before mitosis. So these cyclins, you can kind of think of them as like gas pedals, right? So they're promoting the cell cycle. They're saying, okay, cell cycle, go. And when those proteins get overexpressed, right, they're called oncogenes. So an oncogene is something that's going to promote cell division at inappropriate times when it's mutated. So if these proteins are being expressed at really high levels when they're not supposed to be, it's going to really just drive that cell cycle into overdrive. It's not going to stop. But there is another class of proteins, so proteins like p53 and retinoblastoma protein, whose whole job it is to stop the cell cycle. So these are called tumor suppressor genes. And so they normally will inhibit the cell cycle, but if something happens where they get a mutation and they don't work anymore, right, so their normal job is to stop the cell cycle. So if they're around, they're going to stop it. So we're not going to have that green button going anymore. But if something happens to them and they go away, there's no brakes. anymore, and the cell cycle can continue unchecked. So how do we get there, right? So what is causing these proteins to stop functioning the way that they are intended to? So remember that cell cycle regulators, so all these cyclins and P53 and RB, they're just proteins. So this is their actual structure. And we get protein sequences from your DNA sequences. So this is what is encoding these different proteins. So if something happens to the DNA, if there's a mutation anywhere in the DNA, it's going to end up as a mutation in the protein. And sometimes that mutation will break that protein and cause it to behave in ways that it should not. So what's causing those genes to mutate in the first place? Well, there's lots of mutagens, right? So a mutagen is anything that can modify DNA. And so DNA at the end of the day is just a chemical. And things like radiation can alter that chemical. So UV radiation can break, these bonds between the sugars and the phosphates. So you can get that from the sun. Or if you, play around with radioactive spiders or something like that, you're near a nuclear site, that's going to cause breaks in the DNA, which will cause some chromosomal arrangements. You might encounter a chemical, some mutagens that alter the DNA sequence itself, so the nucleotides, which is going to cause a change in the proteins. So you mutate the DNA, you're going to mutate those proteins. So carcinogens are a specific class of mutagens. So carcinogens are mutagens that cause cancer. And there are some well-known ones, so probably the most infamous is cigarette smoke. Some viruses can also be considered carcinogens, and like that black stuff that you love to eat at burnt barbecue stuff, that is also a carcinogen, it grieves me to tell you. So other things like mercury are very well known to cause cancer, so basically you should avoid all of these things if you can. Okay, so once there is a change in DNA, and you have those proteins no longer regulating the cell cycle, those cells will start to divide out of control and they'll form something called a tumor. So a primary tumor, just like the name suggests, is something that happens first, right? So this is the site of original cancerous cells. They don't move, they kind of stay in one place, right? So these are considered to be benign for the most part, because you can just sort of take them and snip them out and separate them from the person, and that person is going to go off and live their life and be totally fine. So these are the primary tumors. They're not too worried when they see these. But the problem happens when there is a metastasis. So metastasis is what happens when cells from that primary tumor start to break off and start traveling to other parts of the body. Because once those metastases are there, and again, it only takes one cell to do this, they can start to form secondary tumors, right? Because these are cells that are going to divide uncontrollably. So once they start forming tumors at these secondary sites, those metastases are going to start causing problems for the person as a whole. So as more and more tumors start metastasizing, they separate and form secondary tumors all over the person. And this is the part of cancer that is considered deadly and dangerous. Once that starts to happen, it can get into tissues like your brain or your lungs, which you obviously need to breathe, or any, in your midsection, start interfering with your nutrition. So when people catch tumors in their primary, they don't tend to worry about it too much, but once it starts spreading to neighboring tissues, then they start getting a little bit nervous, because that's really the point at which that disease becomes much more serious. So the most infamous, I guess, and deadliest kind of cancer is lung cancer. So this causes more deaths than any other kind. It affects men more than women, probably because they are more likely to be smoking cigarettes, right? But you can also get it from being exposed to radon. We don't really have that problem here in the South. This is more of a Northeast thing. Or asbestos. If you've ever seen people cleaning out old buildings, they're removing that asbestos because it does cause lung cancer, right? So this was not always known, although it has been known for a long time. So studies in the 1950s people started to figure out that there was this huge increase in lung cancer. So prior to 1900, smoking cigarettes was not that common. So the rates start to kind of go up, especially after World War II. They keep coming up. But about a decade later, they start noticing this really rapid increase in lung cancer. And it wasn't until about the 1950s that the scientists figure out that there's this connection between smoking cigarettes and getting lung cancer. But again, look at this delay. You're looking at like a 20-year lag time between, how long somebody's smoking and when they're actually developing cancer. So the scientists go and run and tell the cigarette manufacturers, like, hey, your product is killing people, it's giving them lung cancer. But they took that information and they basically tried to bury it and continue to sell their products, knowing that this was causing lung cancer in millions of people. So these are actual advertisements from the 1950s. So as that data started to get out, they started trying to say, like, well, our cigarette brand is safer. Our cigarette brand, doctors use it more, so you should too. So let's take a look at this one here, because they're never take candy from strangers and never trust corporate scientific studies. So this one is saying a medical specialist is making regular bi-monthly examinations of a group of people from various walks of life. 45% of this group have smoked Chesterfield for an average of over 10 years. After 10 months, the medical specialist reports that he observed no adverse effects on the nose, throat, and sinuses of the group from the smoking Chesterfield. And they're claiming that their product is healthy. But if you are looking at this, poke some holes in this. So first of all, he's saying that these people smoked for 10 years and that he's observing them for 10 months. Well, we know that there's a lag of 20 years, so it would take about 20 years of exposure before you start to see the effects. And you're definitely probably not going to catch it after 10 months, on top of which he's not even looking at the lung. They don't even try to make that claim. They're just looking at nose, throat, and sinuses. And it's their one doctor. And I noticed that they don't say how many people. It could have been 4 people. It could have been 3 people. They don't say. So yeah, don't trust these guys. So it wasn't until 1964 that the Surgeon General kind of blows this whole thing up. So this is like 10 years after the first reports start coming out that there's a link between cigarettes and lung cancer. He writes his report, Smoking and Health, because at this point in our history, about 42% of adults in America were smokers. And the following year after this report, that's when they finally started requiring labels that you might see on cigarette packages even today that they cause lung cancer, so that consumers would at least be informed of their risk. And after that, there started to be more laws enacted banning the sale of cigarettes to minors. So Texas did not jump on this bandwagon until 1989. So your 5-year-old kid sister could have legally gone to the corner store and bought cigarettes until 1989. So as smoking rates have decreased, so again, we see in 1964 when that report was first released that the rates of smoking have since gone down, thankfully, in the past couple of decades. And then with that, so too, again, with that lag time, that lung cancer rates have also started to decline. So that is it for 1.6. Let me know if you guys have any questions. Just email me. All right. We'll see you soon.